definition. So the first definition is autosomal inheritance. So when you study genetics, you will definitely come across these definitions. So autosomal inheritance is inheritance of genes carried on the autosomes. And autosome is a chromosome other than the sex chromosomes. So sex chromosomes are chromosomes that differ in number between the different sexes. So the only two sex chromosomes in humans, we have X chromosome, which is present in both males and females, and we have the Y chromosome, which is present only in males. So what are genes? Genes are discrete units of genetic material. Okay, so it's hereditary material passed between generations. And genes are located on chromosomes. Okay, so before a cell enters division, it must replicate or duplicate its DNA, and that occurs during the interphase and specifically in the S phase. So that's when the DNA is duplicated. And what happens is that the chromatin is composed of DNA and associated proteins called histones. These are special proteins and they serve a very important function in the DNA molecules. So before a cell enters division, the DNA replicates, and then what you see during mitosis or meiosis in the prophase stage, you see that the chromatin condenses into visible chromosomes, so it gets very thick, and that becomes thick when the DNA molecule wraps around these special proteins, and it just condenses to form visible chromosomes, so they're easy to observe under a microscope. So those are genes, and we'll talk about alleles. Now, allele is a particular version of a gene. So for example, a gene may be a gene for flower color or a gene for eye color. So a gene for flower color may have two different alleles. So most genes have more than two alleles. Some genes have two alleles. So the gene for flower color may have allele for purple flower color, and it may have another allele for white flower color. So these are called alleles. It's a particular version of a gene. So next, let's talk about genotypes and phenotypes. So what's a genotype? Genotype is the genetic makeup of an organism. It's a pair of alleles for a particular gene. So a gene has a pair of alleles, and mostly in all the genetic problems you will be dealing with one gene or two genes, and each gene has a pair of alleles. And a phenotype is a visible characteristic in an organism, so something you can observe, you can actually see with your eyes. So phenotype maybe you can observe purple flower colors as a result if you observe in the offspring. It may be long wings or short wings. You may be able to observe if the color has changed or not. So when two parents are crossed, and if you look at the offspring, you can see what color they are. So that's a phenotype of the offspring. Next is homozygous and heterozygous. So homozygous organisms have two identical pair of alleles, okay, for a particular gene. So two identical pair of alleles, meaning they have the same allele for the same gene. For example, a homozygous organism may have a gene for flower color, and since it's homozygous, it will have two identical alleles. So both of the alleles will be, let's say, purple flower color. So both of the alleles will be for purple flower color. Heterozygous, on the other hand, the organism has two different alleles for a particular gene. So an organism may have, for a flower color gene, one allele may be a white flower color allele, and the other one may be a purple flower color allele. So white and purple are two different alleles, but they're for the same gene, which is flower color. Next is the dominant and recessive allele. So the dominant allele is the allele that is fully expressed in the phenotype of the heterozygote, okay, and it is usually represented by an uppercase letter. So that's the dominant allele. Okay, it's dominant over the recessive allele. Recessive allele is the allele that is not expressed in the phenotype of the heterozygote, and it's represented by a lowercase letter. So you will not be able to observe the recessive allele in the phenotype. The dominant allele will simply mask the recessive allele, and you won't be able to notice. So it won't have no effect on the appearance of the offspring, but the dominant allele will have. Next is true breeding organisms. So in Mendel's experiments, he had crossed true breeding flowers, right? He performed 7 experiments, and we'll talk about the results he obtained in my next video. But true breeding organisms are basically organisms that produce the same variety of offspring over many generations. So a plant that has color purple, it will produce purple offspring, purple flowers as offspring for many generations. That's true breeding or pure breeding, same thing. Next thing is a Punnett square. So what's the purpose of a Punnett square? Punnett squares are basically, it's a very handy thing to have and to know how to use it properly well. So Punnett square is basically to figure out the allele composition of the offspring between a cross. So you cross two parents, okay, and these are individuals of a known genotype. So they have, you know, their genotypes, okay. And the genotypes are the pair of alleles for a particular gene. So the genotypes will be given for the two parents, and when you cross them, you are able to use the Punnett square to determine what types of offspring you would get. Okay, so that's the purpose of the Punnett square, to determine the allele composition of the offspring. Next is test cross. Now, a test cross is only used in very specific circumstances. So a test cross is when you breed an organism which has an unknown genotype. Okay, so you don't know its genotype, you want to perform a test cross, you will have to cross this organism with an unknown genotype with another organism that is homozygous. Okay, so homozygous, it will have two identical pair of alleles, okay, and it's also recessive. Okay, so it's a homozygous recessive individual, and you will want to cross it with the other organism which has an unknown genotype. And by just looking at the offspring, by observing the phenotype of the offspring, you can reveal the unknown genotype of the organism. So that's the test cross. Test crosses, you always have to cross the organism with unknown genotype with a recessive homozygote. Remember that. So we talked about sex chromosomes, chromosomes that differ in number between the different sexes. And last thing is sex-linked genes. And these are genes carried on the sex chromosomes. So these are your genes are mostly located, most of the genes are located on the X chromosome. Okay, very, very few genes are located on the Y chromosome. So basically, sex-linked genes are also called X-linked genes, right? Because they're located on the X chromosome. And these are the major definitions that you will need to know in order to correctly solve the problems. And in the next videos, I will introduce some of the simple problems in genetics, and I will also retell you these definitions as I go along and solve these problems. So I hope you enjoyed, and don't forget to subscribe. Thank you. Bye.